Thank you so much. And first, I want to apologize for being a few minutes late. I guess it's fashionable to be late. It always looks okay. It's not too bad. So I have some conflict of interest just to make sure, I received some funding from various companies that develop stimulants or treatment for narcolepsy, since I've been in the field for many years. So we have a lot of clinical trials, for example, and occasionally I also consult to just help people to design clinical trials. So my story really started with narcolepsy because, when I came to Stanford, I just thought that it was a very interesting model to study. And, but of course, it starts even before me. Narcolepsy is a kind of an odd disorder. It was first described in the 1870s, to my dismay, a German physician, Dr. Westphal, and then a little later, a French physician called it narcolepsy. And in fact, in France, it's called the Gélineau syndrome, whereas everywhere else it's called narcolepsy. The idea was, the debate was that these people would collapse suddenly. So it almost looked like a seizure. Then there is this epilepsy, and then they had this sleep attack. So narcolepsy was a combination of these two elements. In the 1930s, you probably know that the amphetamines were invented. In fact, they were overused quite a bit during World War II in some circumstances. In the 50s, Dr. Dement and Aaron Abrazansky and so forth described REM sleep. And then quite quickly after, it became evidence that some of the symptoms of narcolepsy was abnormal dreaming, being paralyzed suddenly. It looked a little bit like REM sleep. So people thought, oh, maybe narcolepsy is a disorder of REM sleep. And indeed, they found that patients with narcolepsy go straight into REM sleep. So it became really like a REM sleep model. And then in the 70s, again, Dr. Dement, my predecessor, was here. He discovered dogs with narcolepsy by complete chance. He was talking to some veterinarians, and vets told him, oh, I have these dogs with the same problem. They fall down all the time. And when they're excited, as you will see, that's one of the symptoms. And in addition, they sleep all the time, which of course you can imagine for a dog is much more difficult to objectively see, because I have a dog with narcolepsy and I had dogs without narcolepsy and they sleep all the time anyway. So, but then in the 1980s, there were a little bit of surprise. People discovered that this odd disease in Japan was associated with HLA. And then that raised the possibility that it was autoimmune. And people looked for a very long time to just prove that hypothesis. It didn't look autoimmune because you develop it when you're a child or an adolescent, and then after you have it for life. So it's not like, remitting, relapsing, and actually all the trial of immunosuppressant have been, failed, but we'll discuss that a bit later. But still, there was this weird HLA association, and I worked quite a bit in HLA, and I could show that at the end, it was not the initial factor that was HLA-DR2 that was really associated with narcolepsy, but another gene close by called DQB10602, which is another HLA gene. And I'm very proud of that study in the 90s because it worked using African American. We could show that there were different haplotypes, and it was kind of genetic mapping through cross-ethnic studies. And then in 2000, as you will see, I work on this dog with narcolepsy, and there were one model of this dog had an autosomal recessive transmission. So I cloned the genes that cause narcolepsy in dogs, and then I found it was a receptor for hypocretin, and then we discovered that the cause of human narcolepsy is different. It's a lack of orexin itself. So after we've been working quite a bit on autoimmune basis of narcolepsy, and then the broader spectrum of what is narcolepsy and what is not narcolepsy. And then now there is, as you will see, a lot of different things that are coming in terms of new treatments, which I think are going to change quite a bit the way we treat narcolepsy, but also probably the way you treat some other neurological disorders. Yes. So this is a typical Italian patient. I collaborate a lot with an Italian group in Bologna. Cappuccino is perfect there. And you see one of the symptoms that's very odd is when patients get emotionally excited, they get paralyzed, and they may even drop completely paralyzed. They're awake. It's a little bit like REM sleep paralysis. And the patient remember everything, he hears everything. Sometimes he would try to move a little bit, as you see, but he can't. He's completely weak all over. It's unusual that they will hurt themselves, so it wouldn't be like a seizure, because usually they have time to hang on into something. And then what's difficult is very often they are partials. You rarely see it because it's triggered by he was making a joke, probably politically incorrect, in Italy, and then he kind of collapsed, and that's very typical. Usually in patients with narcolepsy, cataplexy is mostly triggered by laughing, joking, and it's really humor. It's very odd. It's when something is funny for the patient themselves. And often it's more in the context of family members. So when they see people that they like, etc., it's much harder if it's in front of a doctor, for example. So very often you don't see it. And sometimes it's very mild. It's from these heads that drops. But if a patient tells you, every time I can't tell a joke, if I tell a joke, I can't really talk anymore, you know that he has narcolepsy. There's very few, we can discuss that. There are rare neurological disorders that can have secondary narcolepsy, but they are really very rare versus narcolepsy. In addition, they are very disturbed nocturnal sleep. They don't sleep well. They dream a lot. Sometimes the dreams are so real that they think that it happened. For example, typically I had a lot of patients that say in the morning, they wake up and they're absolutely certain that someone broke into their house. And they check, my God, I mean, the window is not broken, but I'm absolutely sure that someone came and broke into my house. So they're very vivid dream that looks almost real. And indeed, to diagnose it, the classical test we do is we bring them to a sleep lab, we study their sleep, and then the morning after, we do these little naps every 2 hours. And then a normal person like you, maybe not the residents, but wouldn't fall asleep or fall asleep very slowly, in 10 minutes or so in these naps as a mean for 5 naps. But a patient with narcolepsy, it's just completely abnormal. They fall asleep immediately. And also they go straight into REM sleep. So they immediately would dream. And often you have 2 or 3 naps like that when you observe REM sleep, whereas normally REM sleep really occurs in the middle, you probably know, in the early morning hours or after 1 hour or 1 hour and a half after falling asleep. And that's very abnormal, it's typical. Unfortunately, you will see that this MSLT is a good test, but it has been abused. And as you will see, as a lot of cases, sometimes a disease has been created by the use of this test, which is called narcolepsy without cataplexy. But for narcolepsy with cataplexy, it's a really good test. It's 95% specific, 95% sensitive. It's quite a good test. Of course, you should never be blind with a test, but still, it's a pretty good test. But unfortunately, it has been done in a lot of patients without cataplexy or less clear sleepiness. And now there's an epidemic of patients that are diagnosed with narcolepsy without cataplexy. And you should you all know that the test is, you see that with GAD and all kind of other tests. A test is never perfect. So anyway, it's sporadic. It's affects about 1 person for 3,000. Normally, typically now we treat it with a combination of dopaminergic stimulants, antidepressant because they suppress REM sleep. So they suppress cataplexy, which is this muscle atonia, the bad dreams. And then we give this weird drug, which I'll talk to you a little bit later, that's called gamma-hydroxybutyric acid, that has been rebranded as oxybate because gamma-hydroxybutyric acid is GHB. And as you know, it's known as a daydream drug. It's a very unpopular drug. So they have called it differently. They called it oxybate, but it's really gamma-hydroxybutyric acid. And it's kind of a strong sedative we give at night. And it really helps people to sleep better. And curiously, we don't know why. For narcolepsy, it really helps. They have less cataplexy, less sleepiness, and it helps over time. I will talk a little bit about it because it's a remarkable drug, really. So again, in dogs, we had these dogs with narcolepsy. And the best way to trigger narcolepsy in dogs is either when they play together. Here is, they are kind of playing, it's a litter. And one of them is like already down, paralyzed, like the patient I showed you. Like now they're all paralyzed. So you can get them to play together, or of course, you give them good food, and food really works. But it has to be good food. I have a narcoleptic dog at home. I thought about bringing him. You give him like good food, French cheese, whatever is, it really works. Here it has to be fun. And so a lot of my work, which I won't detail, was really took 10 years, is to breed these dogs together or find these odd Dobermans that had narcolepsy all over the country, get their blood sample. We had an adoption program, and then try to track the genes through these families of dog. And it took 10 years, but then when we discovered it, by the way, it was not always easy to breed these dogs with narcolepsy, but because when they get excited, they collapse. But we still succeeded. We got this very nice result, and that was really luck. You have to say, sometimes science is definitely about being smart, but being lucky is probably more important than being smart. And this was kind of lucky because, as you see, it led directly to the cause of the human disease, and in a very simple way, because it was a receptor for chemicals that just had been discovered 1 year before that was called orexin. And it was called orexin because it was a big times of leptin. You probably remember leptin, the 1980s, and people saw that was a solution for obesity. And then finally, now it's GLP-1, but it was all in the hypothalamus and all these neurons that regulate body weight and appetite in the hypothalamus. And leptin was, no doubt, a big discovery. It's really a starvation response, but it was in the hypothalamus, so people were really focused about the hypothalamus being very important for body weight. And you also know that raw age and some of the hypothalamic pathology are often associated with big gain of weight. So when people looked at this orexin, they found that all the cells that produce this orexin, they are in the hypothalamus. And Dr. Yanagisawa in Japan injected orexin in the CSF, and he saw that the animals would eat more. And then he called it orexin, increased appetite. Unfortunately, it was kind of an artifact because it was working when he was injecting the drug during the daytime, which most people do, but not during the nighttime. In fact, he was waking up the animals, and they had nothing better to do, so they were eating more. And when you were injecting it actually during the nighttime, where they're already awake because they're nocturnal, it was not having such a big appetite stimulating effect. So now it's very clear that it's not really an appetite stimulator, but it's mostly a wake promoting compound. And as you will see now that we have drugs, it's amazingly effective. The orexin agonists are probably as strong as amphetamine, or actually stronger, but they have definitely a different profile. So these orexin neurons are all in the hypothalamus, and there's about 70,000 of them in the brain. So it's a very small number of neurons. They are kind of a little bit dispersed, but they project heavily to all the monoaminergic cells like norepinephrine, dopamine, serotonin, etc. And pretty much in every part of the brain. There are some exceptions. They are stimulatory, so they produce increased firing rate. So the idea, and it also projects directly to cortex and many other areas. But in general, it looks like this is like a little center that itself excites a lot of other brain areas that themselves, also produce even more alertness. So it's kind of a super kind of nuclei that produce a lot of wakefulness. And so it makes sense that when it's missing, you have narcolepsy. And also you have this REM disinhibition, because a lot of these monoaminergic cells are completely silent during REM sleep. And for example, when you give antidepressants, you increase monoaminergic tone and it suppresses REM sleep. So it kind of makes sense in terms of how it works. It's kind of a group of cells that stimulates a lot of the monoaminergic tone. But it probably does a lot of other things I want to make clear, and it's a simplification. It's also probably doesn't regulate only sleep, but other things. The only thing is the sleep is the most kind of obvious phenotype when it's missing. One thing that's important is there are two receptors. There are actually two orexins, orexin A and orexin B, which are also called hypocretin 1 and 2 in the literature, which is confusing because it was discovered at the same time by two researchers, but I think orexin is now the name that's being used by everyone. And orexin A and orexin B are two very similar peptides, just a few sequence difference. And there are two receptors, and to make it complicated, one of the peptides has an affinity for both, and the other has only an affinity for one. And the one we found that's mutated in dog narcolepsy was this orexin receptor 2. and the orexin receptor 1 was known. But it's very important for you to know that there are two receptors, because right now, all the drug companies have really focused only on one receptor, this receptor 2, and I will tell you more about that. The reason is that the receptor 2, the one we found is mutated in narcolepsy in dogs, is kind of a dominant receptor. It's the oldest, it's present even in bird, fish. I even studied orexin in fish, and they have like 12 cells that produce orexin. So it's a relatively ancient system. But in mammals, it has been duplicated, and there's receptor 1 and receptor 2. And they are both very expressed massively in the brain, but the receptor 2, for example, is a little more on histaminergic neurons. You know that histamines is also a stimulant, and then when you take antihistamine, you're more sleepy. Whereas receptor 1, for example, is a little bit more in the locus coeruleus, for example. So it may maybe act more on adrenergic system. So, and there is some evidence from animal studies that orexin receptor 1 could have some self-administration property. So people have looked a little bit away from receptor 1 because they're worried that it could be addictive. Whereas receptor 2 drugs are not, self-administered. But definitely the receptor 2 is more important because if you mutate it, they have narcolepsy, cataplexy, the animals, like the dogs. Whereas if you mutate only receptor 1, they are sleepier. They have a little bit more REM sleep, but it's not like the full phenotype. And in humans, when you do like genome-wide associations of sleepiness, you do find receptor 2 polymorphism that are associated with sleepiness, and receptor 1 polymorphism are also associated with sleepiness. So I just want to point out that both receptor 1 and 2 are probably important, but receptor 2 is dominant. And for many reasons, people have been really working only on receptor 2 for now. So of course, when we discovered this very exciting results of the receptors, that's where I was completely lucky, mutated in the dogs. And at the time, you have to realize a lot of people didn't even believe that these dogs that I was working on had narcolepsy. I mean, I had a lot of like depressing dinner with some people who told me I was working on an unknown neurological disorder in dog. But anyway, at the end, it turned out that it was directly, applicable to human narcolepsy. And again, that's luck because, like in Parkinson, you find tons of genes and they don't give you the solution for all of Parkinson. So, but here it was really simple because in humans, it was really the orexin itself that was missing, which made sense because you have the HLA association and this autoimmune kind of potential process. And then it develops, in childhood so it fits very well with an autoimmune disease. The only thing, as I mentioned, is it doesn't react to immunosuppressant. But the problem maybe is that once you develop narcolepsy, you have all your orexins pretty much gone. When you have narcolepsy with cataplexy, it's all gone. And if you stain the brain of patients, I mean, they live a normal life, but some of them have donated brains. And we stain for both peptides. We measure the peptides content. We looked at the mRNA, which is here. And you see that the cells that produce orexin in the perifornical area of the hypothalamus are all seems to be missing. And the cells next to it, like the MCH cells, it seems very normal. So it seems like there's a very specific loss of orexin. So a lot of people have asked, is it like just the orexin that's missing, or is it the cells that are destroyed? I think there's like 99% chance that the orexins are destroyed in the sense that we have tried to co-stain them with other cofactors that are also expressed in orexin cells like NARP, dynorphin, and when you look, the signal is gone. So it looks like there's nothing that produce what usually orexin cells are producing. There is a minority opinion that thinks the cells are like shut down but not dead, and it's like demonstrating the presence of a zombie. I can't really be absolutely sure, and the only way we will be able to really know that is when we succeed to do single cell sequencing in this region of the brain, for example, which I don't think is going to be, I don't think the technique is yet, good enough to show that maybe there's this remaining cell group without maybe the orexin and some other cofactors. But I think so most likely it's really gone. And that fits very well with the autoimmune process. So when we discovered all this, one thing that happened is narcolepsy became much more famous. Nothing is better than finding something in dogs. It's very popular. And we started to see kids with narcolepsy. Before, we had never seen kids with narcolepsy. They were always like people who had narcolepsy for years, and they didn't even remember when it started very often, because often it started around 7, 10 years old, and sometimes they really forget when it started. And we realized that narcolepsy in dogs is a little bit different. In humans, young kids, is a bit different. It's very dramatic, and often you have this general muscle atonia. So like this is an example. In China, they almost have a general loss of muscle tone. It's much more dramatic. And then they often have a lot of grimacing, and they have these tones that move forward like that. And it's not always triggered by emotion. It can be like a little spontaneous. And often they gain a huge amount of weight suddenly. I mean, they can gain like 10 kilos in a few months. So it's very dramatic. And in fact, they often even develop premature puberty when they're very young. We don't know if it's because of the gain of weight, but it can be really dramatic. But what's nice is you can really time it. Often the mother will tell you, oh, it started exactly this particular month. So we started to see more and more of these kids. And then at the same time, and I'm not going to go into the detail because I don't have time to really talk about everything, but I started to work with China, and when it was still possible. And what's good about the Chinese, there are a lot of Chinese, so they have a lot more narcoleptic. And in particular, I worked with this person in 2004, and very nice, and he started to be interested in narcolepsy. We did a little news in the web, and then a lot of patients were diagnosed. And curiously, he got mostly kids. I suspect that it's because of the one-child policy, but he really got a lot of children with narcolepsy that were diagnosed, and now I think there are like 10,000 in China that have been diagnosed. It's funny how one meeting can make a big difference. And we quite quickly realized that it was kind of funny. At the same time that I saw these kids, I realized that a lot of them had sore throat. Often they developed some of them even had developed like something like PANDAS, it were like misdiagnosed, and almost sometimes even chorea, because they have, when they are paralyzed, they often have movements that look almost choric. I don't think it's chorea, but they try to fight it, because of the atonia, and can look a little ballistic. So they were like this overlap, a little bit with chorea. And then I thought initially that it was strep. And we found that a lot of patients with narcolepsy had indeed like high level of ASO. And then you will see, we discovered, and then at the same time, we started to plot when patients start narcolepsy when they are kids, because you can really tell exactly the week. I mean, it's amazing. It's really like the parents can tell you, whereas in adults, impossible. It's often very slow, and you don't really know when it started. And you see that it was very clear. It was always a peak, during the summer or the spring. And then look what happened in 2009. Then I changed my mind, of course, because in 2009 was the swine flu. And we found this huge peak of like a lot more patients with narcolepsy developed it after the swine flu epidemic. And at the same time, there were like a vaccine that was developed in Europe that was called Pandemrix against the swine flu. And a lot of kids developed narcolepsy after this particular vaccine. We still don't really understand why, because this vaccine was adjuvanted very strong. And the regular vaccines that we got in the US, which didn't have this strong adjuvant, didn't trigger narcolepsy in an obvious way. And by the way, I'm totally pro-vaccine. I mean, I've been vaccinated, there's no questions about that. But it's just like everything. Unfortunately, this debate, sometimes they become crazy because there is the for and against, and anything in between gets washed into the dustbin. But the truth is that vaccines are effective. Because they're effective, they can cause side effect. I mean, I'm a pharmacologist by training, and you say no effect, no side effects. That's the only solution. So, and here, we have these cases that were really triggered by this vaccine, and it's very well established, like 10 times increased risk, 10 to 15 times. And there were an epidemic of like young child developing narcolepsy after this vaccine. So it really became evident that it was the flu that was related. And indeed, at the same time, because we couldn't find the autoimmune process, we really look, and I'm still looking for it. I'm persistent, but we have not, some people will still, debate, is it autoimmune? The reason is that we haven't found any autoantibody. Most autoimmune disease, it's relatively easy. You take a blood sample and you find out autoantibodies that will recognize, for example, insulin-producing cells. So we did the same thing. We sliced the brain of mouse and rats, and that's the way also a lot of these anti-NMDA and all these autoantibodies have been found. We kind of put the serum of patients on it, and then you put an anti-immunoglobulin, human immunoglobulin, and normally it stains a place. And if you could have seen it stains the orexin neurons, I would have been retired already. But unfortunately, nothing stains. We cannot find any autoantibodies. And we did all kinds of experiments, and everything has been negative. I'm still searching because it's so striking, this HLA association, and there's a loss of orexin neurons, and you will see the genetics is totally autoimmune. It just makes, it's 1 + 1 = 2, but we don't have the equal, So it's really annoying, but I think there's pretty much no doubt that it's autoimmune in some fashion. Indeed, it's triggered by the flu. I mean, it's actually more clear, in fact, than MS and EBV. In fact, recently we even had a new study where we took recent onset narcoleptic patients and controls of the same age, etc., and same season, and we measure autoantibodies against the flu, and we found increased titers of H1N1 and actually also influenza B, which is kind of very interesting for us. We think there are certain flu that trigger narcolepsy and not others. So we're definitely narrowing down how maybe the flu triggers narcolepsy. There's absolutely zero doubt. And then the genetics fits completely. You have this huge HLA association, and then the other genes that are involved, they are also associated with type 1 diabetes, for example. There's a big overlap of genes with type 1 diabetes, which is autoimmune and makes sense. And in addition, there is also very strong association with certain polymorphism in the TCR receptor gene. That will have to go into heavy immunology because the T cell receptors, normally, it's a gene-like antibody genes. They are not, the diversity of TCRs is not mediated by genetic. It's mostly mediated by this somatic cell recombination. I don't know if you remember vaguely, but you have this reassortment of the loci inside B cells that produce different diversity of antibodies. And the same way you have reassortment of the TCR alpha and beta that produce different chains that then combine together to do an infinite number of TCR that then are selected through the immune response from birth. So normally, the TCR diversity is mostly is mostly due to your environmental factors. And in fact, if you sequence TCR, your T cell receptors in the blood of two twins, there's a tiny bit more concordance, but not much. It's mostly like as different as two different people. So it's very surprising that you find actually, very big effect with the T cell receptor. And it really indicates that probably narcolepsy, there's this HLA that's very important. It's 100% DQB10602, and probably a very small set of T cell receptors that can interact with an antigen to produce an autoimmune response that then kills the orexin neurons. I mean, that's kind of very clear for the genetics, but the problem is we can't find exactly how it works. And probably the reason is that it's very specific. Again, the TCR, maybe only a few TCRs. And whereas in type 1 diabetes and all these other diseases, we have the opposite problem. It's more very broad. So you cannot ever find what's beginning it. Like if you look at type 1 diabetes, you have antibody against insulin, against surface cells, against GAD, against blah, blah, blah. And it's very difficult to know what is starting it, because once it's started, there's probably some kind of generalization, and then it becomes very difficult to see what was the initial trigger. And also probably some of these autoimmune diseases have different ways of starting. That will be a longer story. But here's probably a single HLA, very specific trigger, in my opinion, very small set of TCR that then triggers an autoimmune response. But because it's so specific, we have trouble finding what is really happening. But I'm hoping that when we find it, it's going to be more revealing. But of course, hope springs eternal. So we, besides this autoimmune, and I'm not going to go in detail because we do a lot of work in this area. In fact, we have started to do more work in also autoimmune encephalitis and some also autoimmune disease of the brain, because since its discovery in narcolepsy, I think there's an explosion of interest in autoimmunity in the brain. It's very clear that there's many more autoimmune disease of the brain. But interestingly, all of those are being found with antibodies. This one doesn't have autoantibodies. So it still raises the possibilities that maybe, we're missing a series of autoimmune disease that may be more related to T cells, because that would be the hypothesis that maybe this disease is purely T cells. The other possibility is I'm still missing the autoantibody, and it's just a matter of finding it. And I'm open to either. I just don't know, but I'm sure it's one of those two. So because of, another thing that has happened is, as you will see now, there's a new treatment for narcolepsy. So people became very interested in diagnosing better narcolepsy. So how do you go about this? So currently, it's this test, which I will tell you is not a very good test, where you ask people to sleep, and then you do the nap test every 2 hours, and you ask them to try to sleep. And then if they go into REM sleep, like 2 times the following days or more than 2 times, you know that they have narcolepsy. But truly, when you see the symptoms of narcolepsy, it's more complicated. It's not just that they go into REM sleep. Sometimes they are half awake and half in REM sleep. So at the same time, because I had a friend who was doing machine learning, and to be honest, deep learning at the beginning, I was like many other, that's like 7 years or 8 years ago, I was a little skeptical that it was not scientific, but it worked. So we developed a network to score sleep stages, and it works really incredibly well. You feed a network to recognize different sleep stages. We have tons of data. It works actually better than humans. I'm not going to go in details, but what is very nice is not only it works as well as human, but it gives you a probability of each sleep stages. It gives you, like, if it instead of telling you, oh, this is awake, it will tell you, oh, the probability of being awake is 90%, which means that if you have 100 scorers, they would all score, wake. Whereas here you see there's this little peaks here where there's stage 1, which is in red, where the probability of stage 1, so maybe the patient started to, that's the control, this one, like started to fall asleep a little bit, and 50% probability of being in stage 1 and kind of having a little nap, a mini nap. And then here's a patient fall asleep and is going to stage 1, which is red, and then stage 2, and then stage 3, which is blue. So there's a probability. It doesn't tell you it's stage 2, stage 3. It tells you it's 90% stage 2, 90% stage 3. And then after you have REM sleep, that occurs. And here in this case, REM sleep was maybe not completely clear. And that's why the probability is about 50%. And probably if you were doing the same recording with 100 scores, you would probably get we know we'd get a very similar picture. We actually have done these experiments. Look what happened in narcoleptic. It's kind of amazing. Yes, they go into REM sleep very quickly. Not all of them, actually, 50%. But what's very interesting is they have these stages where the computer gets completely confused. It can't say, is it REM sleep? Is it wake? Or is it half asleep? And that's really what patients experience. They are dreaming, but half awake, or they are paralyzed, but they are awake. So they have certain aspect of REM and certain aspect of wake. So of course it confuses the network, which actually patients with narcolepsy can be difficult to score with humans. And it gives you some kind of weird stage, which is half awake and half in REM sleep. So we had the idea of using this and use this probability of, we call that hypnodensity now. It's kind of a probability distribution of sleep stages to try to use that, to then use that as feature to diagnose narcolepsy. And now we can really diagnose narcolepsy type 1, only type 1, but incredibly well with just a nice study by using this probability distribution and using, it's almost like a lasso. We can really, so we are trying to use this to try to screen for narcolepsy in patients with sleep apnea, or trying to use devices. Now there is possibility of doing EEG at home. Actually, you have been doing that for decades with seizures. I don't even know why we don't do it for sleep, but that's another issue. But I think the future is going to be, people being able to wear that at home and then doing machine learning and find their sleep has all these weird characteristics, and then we can diagnose them much more effectively. And of course, I'm trying, in fact, there are many different ways, but now I'm even trying to use actigraphy and maybe a combination of a few questions. Like I've worked with the UK Biobank, And they have now questions about narcolepsy, which will be released probably in January. And then we'll have that plus actigraphy and plus genetics. So we are hoping that with combination of devices and genetics and questionnaire, we might really be able to screen very large number of patients and find the narcoleptics. Emmanuel During, who used to be here, and Mitch now does a lot of this work on RBD, also did some studies that showed that actigraphy plus some questions about REM behavior disorder could be quite predictive of RBD as well. So I think the future is definitely probably not sleep itself, even so it's feasible, but more like devices together with some questionnaire and a method that's called multi-model learning, because the big advantage of using different modalities is false positive have different origin. Like when you ask questions about cataplexy, there's always a crazy person that says, yes, of course I have it, but then the probability is that that person says, yes, I have it, and then that the sleep or the actigraphy will look like narcolepsy is much lower because the errors are of a different type, and then the genetics. So I think using different modalities really increase a lot of specificity. So we are, for example, trying to do this in the context of the UK Biobank to find narcoleptic patients. So in addition to this, as narcolepsy type 1 was discovered, the MSLT was a key diagnostic criteria. So people started to find these patients who had an MSLT that looked like narcolepsy. They will have a few REM onsets, and then they didn't have cataplexy. They didn't look like narcolepsy, in fact. But they didn't have sleep apnea. They just seemed tired. So people started to call them narcolepsy type 2. And when I started, it was acceptable because let's say that 90% of the narcoleptics had cataplexy and 10% were kind of this borderline. But now we do MSLT like crazy. And we do them to everyone that's moving. So if I do an MSLT in all of you, I'm going to find a few false positive because there's 4% of false positive. We already know that. We have done some studies of hundreds of normal people. There is 2 to 4% of false positive, which is a good test. But if you do it systematically, considering that 4% is 100 times higher than 0.03%, obviously you're going to overwhelm the system. And that's what's happening now. We have all this narcolepsy without cataplexy, which to be honest are completely meaningless. In fact, I will show you more about that. And of course, we also find people who fall asleep quickly and are tired. I'm not saying they have nothing. We have patients who complain of being tired, and that's an horrible problem in their life, and they have symptoms, but I don't think it's related to narcolepsy type 1. And some of them also sleep a lot of time. They have trouble waking up. It's very different from narcolepsy because narcolepsy type 1, actually, normally, they have very poor sleep. They have a lot of dreaming. And if they take a nap, they feel better after the nap, but it doesn't last long. Whereas these patients often, they say they sleep all the time. I'm tired. I cannot wake up. I have brain fog. Very different. And we call those idiopathic hypersomnia. And then there's even people who complain of being tired. And then when we do a sleep test, it's completely normal. We don't know really what to do about those. And then there's this very odd disorders that I like, which is on the border of neurology and psychiatry, which is called periodic hypersomnia, which is these people, they're normally young kids or adolescent male, and suddenly they sleep for 2 weeks continuously. It's terrible. It's not like just they have trouble waking up in the morning. Usually they go to the emergency room because often they are even confused. They cannot talk, they are withdrawn, they feel they're in a bubble. And sometimes even you do the EEG, you find EEG slowing. So it almost looks like, confusion. And they go to the emergency room, they do all kinds of things, nothing. So they send them back and then it gets better. So people are very happy. And then 2 weeks, 3 weeks, a month later, it restarts again. And it lasts a week, 2 weeks, and so forth. So it's called periodic hypersomnia. And it's very unusual. I see quite a bit. And we have done some work now, which is quite interesting with genetics. I've collected. I'm a big collector, so I take blood sample everywhere. I'm known as an international vampire, so be very worried about this because I like genetics, and genetics is blood sample. And we we got 900 cases now, and we found that the top gene is actually TRANK1, which is a gene for bipolar disorder. And it's known that this condition often react well to lithium. So it's probably something to do with an overlap between hypersomnia and bipolar. And bipolar, I've been reported to have more hypersomnia when they're depressed. So I think there's some very interesting psychiatric overlap with hypersomnia, periodic hypersomnia, and psychiatric disorder. And in fact, it is kind of supported by some of the studies in the UK Biobank, where when you just look at people who report sleeping 10 hours every night or 12 hours every night, some of the genetic architecture overlaps a little bit with schizophrenia and bipolar. So my opinion is that some of these hypersomnias have a psychiatric overlap, which, by the way, doesn't necessarily mean that they are depressed or anything, because sometimes they don't report depression. But I think it's probably in that area of overlap that is part of the solution. So as I mentioned to you, the MSLT, it's not perfect. We did some study in normal people, and we actually found that about 4%, 3% of the population has a positive MSLT without any reason. A lot of these people have circadian abnormality because REM sleep occurs in the early morning hours. It's very circadian. And so if you're a shift worker, for example, you're going to have a positive MSLT. So never do even an MSLT in a shift worker, because it could be positive just because their circadian phase is completely abnormal. But in general, it was also associated with a little bit of sleep deprivation, but clearly there's a lot of false positive. And now we have taken this narcolepsy type 2, which I told you are these patients who don't have cataplexy. You do an MSLT, they go into REM sleep, but they don't look the same. And we repeated the MSLT in a series of them. And we found that in most cases, it doesn't even repeat. So not only the test is a false positive, but if you repeat it, it doesn't repeat. So it's not even a consistent finding. So clearly, I mean, in my opinion, but of course it will take 10 years to convince the narcolepsy type 2 is not a true condition. It's just it's a condition defined by a test, by a false positive. I mean, not all of them. Sometimes you find a narcolepsy type 2, and then you do a lumbar puncture and you find low orexin, which is the way I told you we can diagnose narcolepsy type 1. And so there's definitely a little overlap, but it's a minority, and it's definitely overwhelmed by all the false positive. But as interesting, I will show you, at the end, you treat them a little bit the same way, and we have quite good results. So it's a little bit mysterious. So that's the last part of my talk is I will talk to you about narcolepsy type 2 and idiopathic hypersomnia. So at the end, narcolepsy type 2 and idiopathic hypersomnia, there are people who complain of being tired. We don't know why. Sometimes we do a sleep test. We don't find anything. We do this nap and they fall asleep quickly. Very often, the difference is they wake up, they have a lot of trouble waking up in the morning. It's just like horrible, Whereas the narcoleptics, they wake up okay, but they feel sleepy after. And then if they take naps, they nap for 1 hour and they're tired, they have brain fog. It's kind of a different phenotype. As I told you, I think there's probably some overlap with psychiatry, but we don't really understand. I think we have to do a lot more work in understanding these symptoms. And like what I did with machine learning, for example, like looking at what happened with the EEG when they have brain fog, or when they have a sleep inertia, to try to understand it. For example, we're starting to look at local sleep. Even for narcolepsy, we're finding that sometimes REM sleep occurs more frequently in some areas than others. And there's some dissociation, which could explain this dissociated REM sleep event. I suspect that some of those, like sleep inertia, you're half awake, but you may have like slow wave sleep in one area of the brain. That's probably a very interesting area to pursue. So there is a need to better phenotype. The other thing is, I think we have to go back to basic physiology. And I think most of you know that two things that regulate your sleep is how much sleep you got and your circadian time. For example, when you wake up in the morning, you're awake because you just slept. So that's fine. You don't need an extra boost. But normally, as time progresses, you should be more and more tired, and become more sleep deprived and become more tired in the evening. That's not what we experience. Usually, we are even more awake in the evening. The reason is a circadian clock sends you wake promoting signal in the evening that compensate for the fact you've been awake all day. So in the early morning, you're awake because you just wake up, you have no sleep debt. Then in the afternoon, you're awake because you're sleep deprived, but you have a circadian drive to stay awake. Then the first part of the night, you fall asleep because you catch on your sleep debt and you have a lot of slow wave sleep. And the second part of the night, you go into REM sleep and you sleep extra because your circadian clock drops your body temperature and makes you sleep extra. That's why if you go to jet lag, you cannot sleep, in the evening local time, because that's when your circadian drive is high. So it's not like the circadian clock makes you awake all day and makes you sleepy all night. No, it makes you awake in the evening and sleepy in the early morning. And really with all this hypersomnia, we don't know anything about it, to be honest. We don't know if it's circadian. Maybe some people may live in a Tokyo time, that's why they're tired. Maybe some of them, it's because their sleep is never good at night. Maybe some of them don't have a wake drive enough. Like, for example, the orexin will be the case. So I started to think, maybe we should try to address this more physiologically. And to do this, I have used like proteomics, because you probably know that now you can measure like 11,000 proteins. Here it's like 6,000 in 100 microliter. It's an incredible overview of physiology. And indeed, for example, right now to measure circadian clock, it's very complicated. You have to measure melatonin, and people have to spit in a tube for 1 hour, every hour, in the evening until you see the melatonin rise in darkness. So it's very inconvenient. But when you measure 5,000 protein, there are some that peak at different times of the day that are purely circadian. And to do that, it's quite complicated. You have to keep people awake for like 36 hours to make sure it's not contaminated by sleep and wake. And then you see things that continue to, cycle. And this way we find some particular proteins that peaks in the day at different time. And then if one is high, low, the other one is low, we can use machine learning and we can actually deduce the circadian clock. So we have this that we are doing. And indeed, like most of the protein, when they peak, it's in the morning, late morning, or in the early evening, which exactly what makes sense. I told you that the circadian clock is really active in the evening to keep you awake, and the early morning to make you sleep extra. So now we have ways to look at the circadian clock, and we can know if people are like completely abnormal, like live in Tokyo time, or are very delayed. And then we also are starting to develop panels of proteins that reflect acute and chronic sleep deprivation. So my dream would be, I take a blood sample, I write the hours, and I say, oh, you're sleep deprived, chronically, and you have, your circadian clock is abnormal. And this way I would say, okay, I need to give you some light to shift your clock and maybe give you a stimulant during the day or make you sleep better during the night. And indeed, that's the kind of work we are doing. I realized I didn't have so many slides, but I've been too slow. So so I want to mention that really the big thing now is how we treat all these patients with sleepiness. And traditionally, we've been using amphetamines or dopamine reuptake inhibitors like modafinil or amphetamines. But our experience is that it just doesn't work on the long run. It seems like it helps people, but after a while, they just want more. They don't like it that much. They are awake, but like robotic. Then there's some like histamine H3 antagonists that release more histamine. It doesn't work very well, but it's a mild compound. But then there's this real compound that I mentioned, sodium oxybate, which you give at night. It makes you sleep very deeply, and then people feel more rested. And amazingly, it really helps a lot of patients, both with narcolepsy type 1 and narcolepsy type 2. And that's the way we usually treat narcolepsy. When you have orexin deficiency, we definitely use Xyrem, because in addition, they usually lose weight. So it's really amazing. We don't know really why, but Xyrem not only makes you sleep better at night, less cataplexy, etc., but also they lose weight. So it's really a good treatment. But often we have to add, like dopaminergic stimulant or antidepressant for cataplexy, etc. In the past, we were using mostly stimulant and antidepressant, but definitely this strong sedative has really changed the way we treat narcolepsy. And amazingly, for like idiopathic hypersomnia, these cases that I thought were like psychiatric a little bit. Surprisingly, initially in the past, we were really using a lot of these stimulants. They work also, but we are discovering that this sedative, sodium oxybate, really works well. Now I mean, that's one of the things I want you to remember is that change in the last few years. We realized that this drug, sodium oxybate, is amazingly active. I mean, even when you give it in people with idiopathic hypersomnia, it makes them sleep better at night. They wake up more easily and they're more awake. And it can transform the life also of people with idiopathic hypersomnia. And then finally, now there are orexin agonists. And it started first with like a drug from Takeda that you had to use IV. But you see, that was really dramatic because with all the drug we have, the H3 antagonist, modafinil, even sodium oxybate you give at night, or amphetamines, you have this test, which is called the maintenance of wakefulness test, where people try to stay awake and they try to fight. And a patient with narcolepsy, it's almost impossible. I mean, you tell them to do nothing and then try to stay awake, they fall asleep in 1 minute, actually in 1.9 minutes or something even less. And when you get all these drugs, sometimes you raise it to like 10 minutes as best. And a normal person should be able to stay like 30 or 40 minutes. You should be able to stay awake the entire time. So none of the treatment we have work really well. They help a lot, but not enough. Look at what happened with the orexin agonist. I mean, just amazing. I mean, they all kind of went to 40 minutes. They could stay awake for the entire test, like a normal person. And now we have used, so there's several new compounds that are being developed. And what's amazing is one, they work better in narcolepsy type 1, of course, because you lack orexin. So you can use a very low dose and then they are already awake. So about the dose, about 1/3, it already makes them awake. And it's very different from the classic treatment. Sometimes it's transformative. They start new hobbies and all kind of other things. It also works in people with idiopathic hypersomnia or narcolepsy type 2, but you have to use a much higher dose, which is normal because they have orexin. So it's kind of supplementing the normal physiology. Whereas the narcolepsy type 1, they don't have orexin. It's like insulin in the patients with type 1. But you see, it's really amazing. And now there's several compounds. And the most recent one is called TAC-861. And they just completed a phase 2, and they're starting a phase 3. And you see, it's really dramatic. On this maintenance of wakefulness test, they can stay awake pretty much continuously. The cataplexy decreases, the sleepiness, everything kind of improves dramatically, much better than any treatment we have. So for me, there's nothing more exciting. I mean, finally, what I've done like 20 years ago is being realized. And of course, as typical, once something works, everybody jumps on bandwagon. So now we have tons of companies that are developing orexin agonists. So whether or not you like it, you're going to have it because there's like 10 companies who are developing these drugs. And the question is which one will really be the best, but they are definitely waking up patients very strongly, like amphetamines, and they have a different profile. I think people are much more engaged, and certainly for narcolepsy type 1, it really changes their life. I mean, they start to go out again. Before they were just treated, but still restrained in their disease. Now they restart hobbies. It's quite amazing to see that for me. And I want to point out also this sodium oxybate drugs, which is a very old drugs. It's like a sedative that was used, but initially we're using it only in type 1 narcolepsy, it's a state rape drug. But now we are really realizing that for type 2, it's also very helpful and idiopathic hypersomnia. And I'm not sure exactly why, but probably promoting deep sleep in some of these patients really makes a big difference. And before, of course, we tried benzodiazepine, regular hypnotic, it didn't work. So you really need this drug. And it's a very old drug, actually. It was first synthesized in 1874, but somehow it's still several $100,000 to treat it. So now there's a lot of clinical trials. So I'm super busy, like with a lot of different clinical trials, but I think there's a lot of options for patients with narcolepsy. So in general, my next goal, my next goal is to better find ways to diagnose narcolepsy at scale, either with devices or machine learning. I'm hoping for blood tests. I still haven't given up. I work with Dr. Wilson, for example. We try to look for antibodies against, using phage display, etc. And I'm still hoping we can find one day an autoantibody or some blood methods, because that would change everything. And idiopathic hypersomnia NT2 is probably frequent, but it's kind of it's like people who are tired without a reason. And personally, I think it's more interesting to try to understand what the physiology is and try to tailor the treatment to the physiology that's abnormal. Circadian or sleep debt, not enough sleep, not enough wake. I think there's huge potential starting to look in depression. So you have to be very careful because you give a stimulant in depression, they may commit suicide. However, we see a lot of patients, they have been depressed and then you treat them with antidepressants. They're not depressed anymore, but they're still very tired and they sleep all the time. I think those would be very interesting to start to use some of these drugs. Actually, off-label, modafinil and amphetamines are used quite a bit. And probably combination therapy is going to be very helpful, especially oxybate and receptor agonist. And then there's still the receptor 1 agonist, which nobody has looked at receptor 1. So I think it's a very exciting area. These are people who work in my lab, and some do machine learning, others do more like genetic, and some do clinical. They're clinical coordinators because we have quite a bit of clinical trials. So if you have patients with this condition, you can refer them to us because right now, by the way, there were orexin antagonists were developed as sleeping pills, but they have never been successful. They work well, but the problem is Ambien is $2. So nobody wants to pay for $6 for a pill, even if it's safer, unfortunately. But the amphetamine, the orexin agonist, that works because this Xyrem drug is like $200,000, even so it was first discovered in 1874. So it's still very expensive. So that kind of sets a high bar for selling the drugs. So that's why a lot more people are developing stimulants. But still, it's really, I think, due to the problem of our healthcare system personally, but that's a longer area. Thank you so much. And sorry, I didn't realize I would be a bit too long.